Welcome. Can I have you? No? Hello? No?
Push off. Hold on. It's taped.
Oh.
Oh, good.
Okay. Welcome. Sorry to interrupt you. There is some business this evening, and I hope you will come here not to eat, but to listen to our nice talk. We are here today to honor our best students and our long-term members, and that will happen after the talk. I'm Eckhard Helmut. I'm a counselor of the section. I've been over 50-year member. I came in 65, and I'm the oldest one, I think, in the room. Are you a little older than I am? No.
No, they wouldn't admit it.
No, I wouldn't admit it. Okay. Welcome.
Is that microphone on?
Can you hear me now? Okay. Now we start again. Okay, now I hear myself in my ear coming up. Okay, now you are sitting nicely here in this Andre's restaurant, and I'm very thankful that we could get this room, because we lost our first place, and then by accident we got the opportunity to come here. And I like it here, and I think you will like it here too. It may be a little bit strange to see the screen, and you have to move a little bit to see the screen, and if you don't hear the speaker, tell me or the audience to hold the microphone closer. Okay, this award ceremony has a long tradition in the American Chemical Society. For many, many years, we honor the students which perform very well in high school. And for a large number of years, we combined our honoring the students with the Chemistry Olympiad. The International Chemistry Olympiad, where students perform here in the first round and then process to the second round, which is coming pretty soon. And then maybe again, one of us will go up to the training camp in Colorado. And up till now, nobody from Kansas City meets the team, but maybe one day it may happen. Okay, today we have one person here who performs very well, and you will hear it afterwards again. He is good in chemistry, biology, and mathematics, and he performed a few weeks ago at the international competition in Bucharest. And he is close of making the mathematics team. Maybe he will make another team too. We will hear it later. I'm so excited to have all these people here. Now, this evening, we have the talk of Dr. Gray's son. For the last 15 years, I invite my friends. And I try to get Grace to come before, but it never worked out. Now, I'm very pleased that Grace's son is coming back. She worked with us at UMKC from 72 to 74 and was a neighbor in the office of Ron McQuarrie and me. And Ron McQuarrie will be honored a little bit later for 50 years. And we are looking forward for a reunion. It's close to 50 years, right? That we walked side by side. Now, tonight, Grace is talking about botanicals and your brain, or something. How important are the botanicals to your brain? And I'm looking forward to listen to it. I don't know if I should read the whole vitae. It's exciting. She was very successful in Colombia together with her late husband. And maybe you remember him and her giving a talk a few years ago on the French paradox. Does anybody remember the French paradox? Okay, the French paradox is the French people eat all the bad food and drink red wine and live very long. That's a big paradox. And we had a nice meeting a few years ago, but now, today, we will see what is new with the different compounds in many, many different foods. And I think most of you have the chemist and see her birth date and all the other things. I can tell it to you if you want to do it, but I think we should start listening to Dr. Sun.
Thank you very much. Can you all hear me? Can you hear me? Good, good. If not, you can come over here and sit in front of me. I'm so very happy today that I get the opportunity to come back to UMKC, Kansas City, and meet some of my old friends, Dr. Ron McQuarrie and Bob Smith, and I guess I don't know most of you guys now because it has been too long since I've been gone. But I always have communication with people here, and actually Dr. Wong, Rosa, and Chi Ming also has been my close communication people. He is in the biology department. So I'm very happy to be out here, and I was told that I give a synopsis of my research, what I've been doing since the French paradox. So I would like to say that I came 1974 to UMKC. I taught side by side with Ron and Eckhart, and after that, we have a number of years of collaboration since I went to Columbia MU. And since then, I had gone through 40 years of research, and mostly looking at the brain and studying what is going on with our aging brain. And I'm most concerned because I'm getting old. So, yes. So I would like to first give tribute to my husband, who has gone 2 years from before now. I want to tribute him because he is really the first researcher to talk about oxidative stress and talk about mechanism of the oxidative stress in the brain. So in here, I don't have, you can see that me and my husband worked side by side in Columbia, Missouri for 40 years. And the one on the right side is 2 years ago, I was given this presidential award from the system. So it was a surprise reception that I have. So with this, I would like to show one of the slides that my husband showed. This is not my slide. He is always telling me that there are so many molecules in the brain, so many enzymes, so many proteins, but we have to be aware that there's a balance. See, I have a balance. The balance between oxidation and antioxidation. And so today I was told that there are high school students here. Maybe you can learn a little bit. What are some of the oxidative molecules? They all have O there, right? And you can see superoxide, hydrogen peroxide, hydroxy molecule, nitric oxide, peroxynitride. So on the other hand, we cannot forget, if these are all bad things in the brain, we'll be dying in 2 days. But we have something to counteract the oxidative stress. So the antioxidant defense system. Okay, so there are many enzymes and also chemicals like vitamins C and E and oxygen, glutathione molecules. These are the antioxidant molecules. They are very important also in our brain. So they keep our brain balanced. So in my talk, I will be also talking about some of these molecules that are going on in the brain and how can we understand how to balance them. And you can see that, I'm supposed to let you people eat your dinner also. So please go ahead and do your eating while you listen to me. So anyway, there are so many diseases in the aging brain. The aging brain is really, we all worry, especially, for example, Alzheimer's disease. I spent 15 years working on Alzheimer's disease, how we can cope with the disease, how we can delay the onset, and this is, one set of disease that is very devastating. Of course, stroke is another one. My husband died because he had a severe stroke. So it's very important that we understand these diseases that are all related to age. So what in the laboratory that we can do about the aging brain, the diseases in the brain. So some of these compounds are the ones that we have been working. So you can see that about 10 years ago, I gave a talk, and we talked about the French paradox. And that was because we found out that in the grape, there are some goodies. They are in the red wine, and they call resveratrol. And these compounds are very good antioxidant. Of course, nowadays, we all know that the red wine has more resveratrol than the white wine. If you don't know why, you can ask me later. And then we also know that Indian food is good. You eat curry. Curry is curcumin. This is very good stuff. And it has been shown that curcumin or curry has very good beneficial effect for stroke and for other disease. And we also talk about apocyanine. This is from a plant that is in the high Himalayan. They found that this plant is like a herbal medicine. So they took it out from the Himalayan mountains and found the apocyanine, a very simple molecule, but it's an antioxidant by itself. So we also try to study this compound. And at the end, the last one is called the green tea extract component, epigallocatechin. This green tea compound is really very good, and I will talk a little bit more about this. So I'm encouraging you drink red wine and drink green tea. So many years of work came down to these 4 topics. I would like to keep a very short description on each one, because each one will take me a lot of time to talk about the details. So today I won't be able to give a lot of actual data, because I think that's boring in a dinner talk. You don't want to see that. But the concepts that we have been using for our studies. So I will go first. We spent actually about 10 or 15 years on stroke and some botanicals on stroke. So I will talk about that. I will also talk about botanicals that we use for Alzheimer's disease. And then in the last 20 years or so, we get ourselves involved in studying microglia cells. Microglia cells, really interesting cell in the brain. So I will talk about that a little bit. And then in the last 5 years, we have a botanical center and some new botanicals that we engage into studying, which is new to me, actually, but it was very interesting. So in the early times that we look at stroke, we found that the stroke model, the gerbil, is a very good model for stroke. Unlike the mice and the rat, the gerbil lack the communication in the brain. So if you clip or ligate the carotid artery here, the gerbil here, and you can stop the blood flow in the brain. And actually, if you ligate one side, you can stop one side. Ligate the other side, you can stop the other side. And you can use one side as a control if you want to do that. So this is a very good model. So with this model, I just want to briefly tell you, if we ligate or clip the blood flow for 5 minutes, that's very short, like asphyxiation. You clip for 5 minutes, the cells in the brain sense the trauma, and especially the cells in the hippocampal area. The hippocampal area, I don't have pointer. You can see that this area is the hippocampal area. The neurons in that area are very sensitive, especially the neurons in the CA1 area, just a little bit part of the brain. You can see that after ischemic insult, 5 minutes, and after 3 days, the neuron come apart. You can see the good neurons, very nice, round, and the ones that are ischemic, already starting to die. They become pygnotic. So we use this model. And also, besides the neurons, the glial cells and the astrocytes get excited or activated after the ischemia. So just to make the story very short, we use this model. We look at how much the neurons are dying, and we look at how the glial cells and astrocytes are activated. And we have gone through a number of studies by Dr. Chin Wang. He was a postdoc from China. And so you can see that over the years, we have looked at resveratrol itself is a protective agent for the ischemia. Dietary grape, we gave them a grape crush, freeze-dried grape that was from the California Grape Foundation. We feed them, and it was good. And we also used the curcumin, the curry powder. The gerbils like curry. And so we found good effect. And also the last study that we even made a extract, and we put that extract in the milk, and we put it in, give them orally to the gerbil. And we want to find out if we give before the stroke, after stroke, and which one will give us better results. And our results show that actually even after the stroke, you give them the extract, is still good, but not as good as if you give it before and all the way down. So we have a number of studies over the years that we look at how different botanicals go through. So now I want to go to the Alzheimer's disease. I believe most of you know what Alzheimer's disease is. It's a very dramatically devastating disease, and involving the neurons, and involving, you can see that the neurons, the Alzheimer's side, the tangles are in the neuron, they call fibrillary tangles. You can also see that in the brain, they are called amino plaques that are accumulating. And there are many hypotheses and theories about how this disease leads to dementia and what we can do with it. So we point our finger a little bit more to the A-beta molecule, which is component of those plaques. And I want to just show you the A-beta molecule is this molecule and you can see that there are the blue side and the red side. The molecule can be metabolized in different ways. And there are 3 kinds of enzymes that we call the secretase. The alpha secretase cut where the little hot dog, right here, and cut to this. And the beta secretase cut this side and this side of the hot dog, and that's bad news. When that is being cut, the A beta molecule came out, and that molecule, we are still, up to today, everybody's still trying to look at why you get that hot dog coming out and they become aggregated and they become toxic. So some of the hypotheses, at least one that I'm believing, is that before it goes to a big aggregate, the smaller aggregate we call oligomer, they oligomerize, maybe 3 or 4, or maybe 5. They are toxic. And so in our study, we use this to be our base. So this is okay, an example of how A beta oligomers can be toxic to neurons and to synapse. You can see these red dots, and the neurites, they destroy them, and also they cause the microglia, the yellow one up there, to be activated, and also the astrocyte. And then they will release toxic or inflammation, molecules that are inflammatory, and further hurt the neuron. So it's a very complicated mechanism. And Dr. Monkey, 2006, in the Science magazine, had this picture that depicts some of the toxic effects of the A beta. So in our lab, we've been using, so we decided that we want to go for, see what the A beta do to the neurons in the cell. So my student, Shilat, isolated these cells. These are cells isolated from the baby brain, actually neonatal brain, and they are neurons from the cortex. And you can see that the neurons are very nice. They have processes. The reason I want to show you here is we also isolated. In the isolation, sometimes we get some astrocytes. So when you publish a paper and you said you isolated neurons, then people will ask, how do you know you have neurons, you don't have other things? So we stain the green one is that we use the stain called GFAP, which stain the astrocyte. So you can see that in a field like that of maybe 100 neurons, we have 1 or 2 astrocytes. So that demonstrates the purity of our preparation. So using these neurons, we found that these neurons are very easily activated. And one of the activators is the glutamate receptor, the inotropic glutamate receptor, which we call, one of the compounds called NMDA. This receptor, when you add very low amount into the neurons, the neurons get excited because it will open up a calcium channel. The calcium channel go open, then it's a bad news because calcium go into the neurons, it will activate many things, including kinases, including other things. And so my student first study what happened to the NMDA activation. It activates the green block, is a protein called NADPH oxidase. The NADPH oxidase is one that responsible for making reactive oxygen species. You can see the red one. So when we add NMDA to the neurons, we get increasing calcium, we get increase also in the ROS, in the ROS. And that's bad news. The neuron will die because eventually the mitochondria will become sicky, and then you will have apoptosis, cell death. Am I doing okay?
Yeah.
Oh, good. Can I walk a little bit more? Good. Okay. So with this in mind, she also then put in the A beta. See the A beta, which is the yellow one, and we were surprised at the time. This is her finding. When we add A beta, we don't have an NMDA. It does something that is similar to what the excitotoxic receptor. It looks like the A beta activated also the glutamate receptor and do some of this. So when she at the A beta, she measured the ROS, it was increased. She measured some of these kinase, it was activated. So that demonstrated to us that there is some toxic A beta in our preparation that activated the neuron. Okay, so with that in mind, my next student, Yan, she took on this to look at what's the mechanism of the toxic A beta. So she keep adding A beta to the cells, and she measured ROS, because I told you an NADPH oxidase is a enzyme that produce from oxygen to reactive oxygen species. So in her study, she also has many, maybe 4 years studying this subject. So we say, okay, if we can stimulate the neurons and these neurons get toxic, and actually the mitochondria is dying, so can we rescue them? So that comes along with the last word is the green tea EGCG. So she add the EGCG, which is the green tea extract, to this side, and you can see that the green tea EGCG can inhibit the ROS and make the neuron more happy. So that was a good paper, and when we published this paper in 2011, we got the most cited in the ASN Neuro. It's a new journal. So we were very happy about our work. So, and Yan went on to become a postdoc in Brown University. So these are very interesting. So what about EGCG? That's from the green tea, extracted from the green tea. So then we also thought about what to do with EGCG, because it's protective to the neurons. So my two colleagues, Dr. Simony and Shackman, Shackman is from the psychology department, and Dr. Simony is from biochemistry. So get together, and they said, okay, let's feed them to the mice and see what happens, because we just did the experiment with neurons. I mean, that's in vitro study. So what happened if we do that to mice? So what kind of mice? We have to do it with an Alzheimer model. You know, mice don't have Alzheimer's disease. They can remember forever. Yeah, but if we add the gene, we transfer the gene into the mice, this is called the APP gene, that we took it from the human mutated gene, the hereditary familial A beta, Alzheimer's disease gene. We put it in there. There are now number of this kind of transgenic mice. They will become Alzheimer-like, and they will forget, and they will have plaques. Before, we cannot see plaques in the mouse brain. Now, if we do the transgenic mice, they have. So, and we can see, we decided that we want to do something fancy. So we want the mice to drink green tea. And we also wanted the mice to exercise. Up there, the mice are doing exercise. And so this is an experiment we just published just a couple months ago. And so I just go a little bit more about this experiment because I think it's interesting. So in this experiment, we use this CRND8 mice. They are transgenic mice, and they are actually these mice, by 3 months of age, we can see the plaques, and we can see behavior deficit. So we can start to do with 2-month-old mice. Then we feed them for 4 months. And in the middle, we do a number of behavior studies. And at the end, we sacrifice the mice, and then we look at the brain. We measure A beta. So there are 2 tests that I want to introduce to you. The first one is called the nest building. Nest building, this is funny. I like this experiment. You see, you make up your bed. Some people don't make up your bed at all. Other people do, right? Well, mice, if you give them a pad, this is a pad of our cotton pad, a little white pad, and within 24 hours, they will shear the pad, and they will make funny, and you can see they build a nest. And at the score of 5, they are very happy. They build a nest, they stay in there, they become happy. And that environment is very similar if you put a male mice or a female. They both do that. It's not only the female one too. So anyway, and then we have this, we score them. So if they make a mess and they don't like it halfway, you can still see the cotton not being all straight. That's score 2. Score 1 is that the mouse doesn't like the cotton. Don't even touch it. That's bad. The mouse is crazy. And so score 3, and then you can see 4, they have built a nest, but not so nice. It's kind of flat, not so fluffy. And the best is score 5. Okay, so we have students, and they come and score them every day, and then do this experiment. And so this is a very good experiment for high school too. So the results. At 2 months, this is a non-transgenic mouse, and these are the transgenic. See, the transgenic mouse, they have A beta in there. They are Alzheimer. They don't like to do anything. So they are actually not good. See, all of them are, I mean, this is 2 months. We haven't fed anything, but we just divide them into groups. But the interesting thing is after 6 months on the paradigm, you can see the control, the non-transgenic, transgenic mice still down here like this. But if we feed them with the green tea, the exercise, and green tea and exercise, they all improve. They all improve. So actually they have the exercise and the combo almost like the non-transgenic. So that's exciting news. So you can improve yourself, and you can make your bed if you don't make it before. So by drinking green tea and exercising. So that's one data. Another data that we use these mice is called the Barnes maze. The Barnes maze is that mice they like to explore. You put them in a circle like that. This is actually a drum with size, and there are maybe some cues, some circles, some triangle cue. And we in there, one of the circle, like down there, has a duck box. And they like to be in the duck box. So if we put the mouse in the middle, just in the middle, and we shine a light in it, the mouse will quickly find something to hide. They would like to find a place to hide. So they will go around, go around, go around, and if the hole has no duck box, it's still bright, they won't go in. Otherwise, they will drop to the bottom. But they will find the one that has the duck box. So this will be another experiment. You can track how long the mice will take them to go to the duck box. And you can see in the latency is the length of time that go to the duck box. And if you do a training, if you do each day, you will know that they all learn a little bit. So by the fourth day, they either Alzheimer, no Alzheimer, they can learn. But you can see the transgenic mice, the red one line on the bottom, on the top, is always worse than the ones that were given the green tea, the exercise, and the combo. In fact, they all come down to like the non-transgenic. That's really good news. And so people in my university are interviewing two researchers about this experiment. And I think there may be some news about this study that we will have. And also error. We also track it, how many times they go to the hole and to find the right hole. And you can also see that the non-transgenic always, this is a non-transgenic, and this is a transgenic. And with the two treatment, they are in between, and they become better. Okay? So I would say that our experiment was quite a success, but this is one PhD, 3 years of work, not easy work. It's very expensive. And then at the end, we take the brain, and we take parts of the brain, and we measure A-beta as an index of maybe the behavior improvement has something to do with the A-beta. And we can see usually here the transgenic mice, the non-transgenic mice don't have A-beta, as I told you, but the transgenic has. And then when we have these treatment, they all go better. So that's the result of this story, and we are very happy and to be able to report it. So now I don't know, maybe I'm always run out of time, so I think I should go faster. Okay, so I would like to say a word about our study with microglia cells. These cells are in the brain. They are the surveying cell. If you knock your head and you have a traumatic brain injury, and microglia cells are the first kind of cells that sense this kind of injury, and they will become activated. And they can also migrate. They can go fast. So these cells are very active. And you can see the cells, if they are in the brain and they don't do anything, those are the left one. And when they are activated, they have more processes. And sometimes when they are very angry, they even gobble up things. And that is the one in the right side. So they can change morphology, they can migrate, they can gobble up things, they can also release toxic or cytotoxic factors, which is something that we don't want. So, but also microglia are not all that bad. Sometimes they are okay. So we got hold of a cell line, a mouse microglia cells called BV2 cells. Nowadays, 10 years ago, we were only one of the few labs that has these cells, but I have given to many labs. Yeah, they all have very good results. And so you can see, this is Vivian. The cells in the dish are round. You don't do anything, they're round. And if you put some cytokine, we call it cytokines, TNF alpha, IL-1 beta, interferon gamma, they become, in 4 hours' time, they change shape. You can see that they have many long processes. In fact, they are trying to touch each other. This long process on one side and the other side, I call it a cauliflower like protrusion. So these cells, you see, this is a cauliflower, this is a process. And so my tag asked to the question, because we add all 3 kinds of compounds in there, which one is responsible for the processes? And you can see here, the TNF alpha, IL-1 beta don't do anything. The interferon gamma is the one that is like the free cytokine. So you can see that it will do the same job as this. And LPS is endotoxin from the bacteria. It also can do something. LPS plus interferon gamma even more. So now we have some information. Then we know what happened when we add interferon gamma, which is a receptor, and it will do something. And also when we add LPS. So during this time, we got the good news. The good news is our university got a center, a botanical center. So when we wrote the grant, we wrote we want to do more work with the microglia cells, how botanical mediate some of these processes. And you can see that we have 5 kinds of botanicals that we proposed that was 5 years ago. And elderberry is one. This is the one that we wanted to do because one of our faculty members is in Springfield. He has 70-some species of elderberry, different elderberry. They are American elderberry. And he has many acres of, he belonged to the extension, MU extension. So so of course we want to make MU an elderberry center. So, and then we also want to look at garlic, and because garlic has some chemicals in there that you don't want to like the smell, the garlic smell, but it is a sulfhydryl compound that has a lot of interest in interacting with pathways. And pecorizer is the one that I suggest, but we did not do much on it because we cannot go to the high Himalayan mountain to collect the samples to do the experiment. So we forget about that one. And then we also, the MU has a soy center, so soy has phytosteroids in there, and one of our project is on that. And the last one is Sutherlandia. Sutherlandia is a African herbal compound, herbal plant, and I guess the herbal is being taken as a remedy for many things, including HIV and other things, cancer. So it's also called the cancer bush. So with these, my project is on to look at the botanicals in cerebral ischemia. But I will not talk about the ischemia now. So during this time, Dr. Chan, he came from Taiwan. He's a professor there, and he told me that he has 2 compounds, and these are from the magnolia tree, and they are from the magnolia bark. You can see the magnolia, the bark is really very good stuff. It's One is called the honokio, and the other is called the magnolo. And then with my student Dennis, they together look into what these compounds will do to the cells. So we have these microglia cells. So first, we do see whether they are antioxidant, and we also see whether they can inhibit nitric oxide. So I put this as first because we've done this kind of experiment many times. We use interferon gamma because interferon gamma was the one that would give us a lot of processes. And also LPS plus interferon gamma is one that other people also use. So we can see that if we use interferon gamma, we can increase the ROS production in the cell. And then when we add the honokio itself, no interferon gamma doesn't do much, but in here, it goes down to almost like 80% decrease at 10 micromolar. And on the nitric oxide, we also add the LPS plus interferon gamma increase the nitric oxide. And you can see a nice dose decrease with the NO production. Now, I want you to remember, these are compounds that we can already purify, good compounds that we bought and we didn't isolate. So, but they are this is considered very sensitive. They are very good antioxidant and So then the second thing is Jinghua is a postdoc, came in and said, okay, I would like to look at Sutherlandia. What do we do? So we got some extract of the Sutherlandia. These are the ethanol extract that we extracted. And we look at ROS, and we also look at NO. These are the same experiment. But look at here, that we need much more Sutherlandia extract to get to the compound, to get to the inhibitory results. So I just want to show you that different types of botanical and different kinds of results, and the potency is very different. And so the elderberry, you can see that this is, we can get a lot, but right now in Missouri, there are companies that sells Sambucus elderberry juice, concentrated. They are claimed to be good for young kids that have cold, or just before you have cold. It's good for that kind of a purpose. And also it's good. And elderberry flowers, people in Europe also use it for medicinal use. So this is a very important plant that we want to explore, because not too many people in U.S. are looking at it. Other people looking at blueberry or other berries, but elderberry is one that they don't use too much, and we don't see it in the market because it's kind of a little bitter. You don't want to take elderberry as a fruit. Agnes helped to look at the ROS and the NO, and we were really quite disappointed because you can see at 100 micromole per mil, we don't get anything. We have to go to 400. And also with the NO, actually we get some increase and then go down. This is called, we call it a biphasic effect. So they are not all the same. They are not responding the same way. So with this, we did the experiment, and not so, and this is the ethanol extract from the pulpmus of the elderberry. So in the literature, we can see that there are many flavonoids, and some of these are very good, not only in the elderberry, also in the blueberry. And quercetin, and the rutin is a glycosylated form of quercetin, and cyanidin glucoside. And these are very popular, common chemicals that are found in the berries. And so we did take quercetin, and we also take cyanidin. Okay, they look very similar. You chemistry major. Here are two molecules that are very similar in molecule. And so we look at how they can inhibit nitric oxide, and we see that the quercetin at 12 is almost complete, 12 micromolar. With the cyanidin, it doesn't even start. And with 100 micromolar, only like 80%. So two molecules, very similar, very different outcome. Yeah, anybody tell me why? Okay, you can tell me why after you eat. Okay, so with this data, we are really excited because they are both very common in the berries. So what we have used those data on is that so far we have looked at this side of the pathway. We look at quercetin and maybe some other things, the whole no-kill. We look at LPS, which is a pathway. The LPS is endotoxin, toxic. And they induce this NF-kappa B pathway, make the nitric oxide. We've been looking at this. And these botanicals like honokio and quercetin, really good inhibiting, and even some of the extract, we see that. But we haven't seen another side of the pathway or the story, and which is really coming up. If you look into the literature, this is now being even more important. This is called the antioxidant pathway. The antioxidant pathway, a little bit more complicated because it has so-called the KEAP1, NRF2, and the COP. These three proteins, they are very important. They go together, and if nothing happens, they are there, and NRF2 is then being degraded by the ubiquitin pathway, gone. So NRF2 is a very important molecule that come to the nucleus, act on this is called the antioxidant response element. And when that happens, it will make antioxidant enzyme. In fact, there are many enzymes that are making by this pathway. So you have here oxidative pathway, antioxidative pathway. And can, like quercetin, balance it, because quercetin is known to inhibit that. Can we say that quercetin also activate this? And now, actually, there are several papers that prove, yes, if quercetin make this HO will come back and inhibit that pathway. So there are many studies going on with this. So we also, in our study, we measure HO, this is called heme oxygenase 1. And we also measure NRF. So when compounds come along, it will break up that complex. The NRF2 will not be degraded, and it will come to the nucleus, and it will activate the ARE. So this is a rather new and rather important pathway. So you can see here the heme oxygenase system is involved in many many signaling and also molecules. So this is probably the most important, and is not yet published. I submitted the paper and by Jihong Chen. And so what we did is, we know that quercetin is very good inhibiting the inos, the NO. We know that. So we asked whether quercetin can also activate the antioxidant part. So here we have two sets of the experiment. One set is we add quercetin to the cells without the endotoxin. And we see that the quercetin can activate the NRF2, you can see it goes up. Actually also HO1. So this is NRF2, quercetin itself, this is the thing, and also the HO1. But if we add the quercetin into the cells that already has the endotoxin, we know that endotoxin is bad to the cells. It do a lot of things. So when we add that, well, would anything happen? And that was the surprising. It will even go further. It will help the cells to produce more HO to fight against the oxidant stress. So this is the difference between the black and white. Is the exciting data that we have. So normally what we can say in this experiment, you can say that, okay, you in normal situation, you can have some quercetin, and you prepare yourself a little bit, increase the antioxidant pathway. Not only HO1, but many other ones are also increased. And so we call quercetin as adaptogen. This thing can adapt to situation. So it will stimulate this antioxidant pathway in normal cells until you feel better, you feel more happy. And then it will further enhance the antioxidant when you are under stress. And so far right now we only use the endotoxin stress, but we want to study more about the other kinds of stress. And so when I Google to find out what's high in quercetin, you can see elderberries. Can you see that? Elderberry is very high. And what surprised me is red onion. Red onion is really good in quercetin. And cranberries, red hot green pepper. I cannot understand that one. Jalapeno, you get so hot and it was good for you. So anyway, many of these botanicals have quercetin in there, and you can see that the companies are getting good in those. And this is about the end of the talk. We also now are looking at two other kinds of botanical. Dr. Robert Smith is back there. He has been coaching us in what to look at and what other things to do. And he went to Brazil, and he came back with bags of these acai. And so he asked, hey, shall we looking at that? And so we're looking at the acai right now. But this is actually this fruit is very used in Brazil. They eat them and do a lot of things with it. But the Indian ginseng is one that I was very surprised. This kind of product is called Indian ginseng because of the root looks like Korean ginseng, but it also has cherry-like fruit. So it's called, has several names, the ashwagandha and a winter cherry. Those are, and this plant is now it has been grown widely in Southeast Asian countries and even in Africa. And it has been now starting to be noticed to be very good tonic. Especially, it caught my eye that is the effect to combat stress, improve learning, memory, and reduce anxiety and depression. So we are right now doing something with it, and we hope that we will get good data. And so at this stage, you can see, the broccoli has a compound called sulforaphane. This earned a Nobel Prize for a chemist who discovered the NRF2 pathway. and because they use the broccoli, and also there are many other ones. So you can see these are from the grapes to the green tea to the apple to the red onion. These are all compounds of fruit that are healthy, and they could. So I always use this as my ending slide. I think you cannot say one kind of a botanical is good for. You have to be generous in looking at a number of them, and some of them are very good. And also, we also added exercise. This is also good. We proved it in our mice, and also reducing stress is also very good. But I don't have the data for that. So, in turn, I have to thank many of my people. Obviously, I don't do those experiments, and but last year in June, my chair, Dr. Hazelbauer, this is my chair. He suggests that maybe to get me out of my department, he will give me a symposium, so that to invite all my former postdoc students to come back if they can. And I think, Bob, you are there. Here's Bob, here's Dee. You attended my retirement symposium. And so we have also my good friend from Poland, and then Dr. Nicholas Basan, who has been a neuroscientist many years, and then my colleague Gibson and Gary. Actually, the three of us, we call the 3G, because Gibson, Grace, and Gary. 3G. We are the three projects of the Alzheimer Project for 12 years. So we worked together for so long. So it was very nice that I have this. So I guess this is the last one. And thank you very much for not having your dinner yet.